Everywhere you look, in the air, in the water, even on your own skin, there are living things far too small to see. They outnumber us, they shape our world, and without them, life as we know it couldn't exist. These are microorganisms. In this first part of the video, we're going to explore what microorganisms are and what the science of microbiology is all about. Microorganisms include bacteria, both eubacteria and archaebacteria, along with fungi like yeasts and molds, protozoa, microscopic algae, viruses, and even tiny parasitic worms. All of these belong to the microscopic world that requires a microscope to uncover. Microbiology is the field that studies them. It's the foundation of modern biotechnology and branches into many areas. Virology for viruses, mycology for fungi, bacteriology for bacteria, immunology for the immune system, and much more, from food microbiology to forensic science. Together, these fields give us a deeper understanding of life at the smallest scale and how it connects to the biggest challenges we face. When we think about microbiology, it really centers on two big themes. The first is basic microbiology, which dives into how cells work, their structure, their metabolism, their genetics, essentially the inner workings of microorganisms. The second is applied microbiology, which takes that knowledge and puts it to use in real-world settings. Agriculture, industry, and health all benefit from it. From producing vaccines to improving crops or designing new antibiotics, applied microbiology is about turning discoveries into practical benefits. Microbiology can be divided into two branches, basic and applied. Basic microbiology is more about understanding. Sometimes we study microbes by type, bacteriology, virology, mycology, protozoology. Other times we study them by process, like microbial metabolism or microbial genetics. And there's also the disease-focused side, looking at immunology, epidemiology, etiology, and treatments. Applied microbiology, on the other hand, asks how we can use that knowledge. It connects to health through infection studies and pharmaceuticals. It connects to the environment through ecology and waste treatment. And it connects to industry, where microbes help us produce food, beverages, medicines, and even engineered products. It's a web of fields, all branching from one common root, understanding microorganisms. Microorganisms are often called germs, but that word can be misleading. Really, they're just small cells. Many of them grow quickly, but not all are harmful. They each have habitats, and they rarely exist in isolation. Instead, they live in populations. Sometimes they move freely through water, and sometimes they cling to surfaces, forming biofilms, those slimy layers you might notice on teeth or rocks in a stream. Within their communities, microbes interact in fascinating ways. Waste from one may harm others, but that same waste can become food for a different microbe. Some compete while others cooperate, creating a balance. It's a reminder that even at the tiniest level of life, relationships and interactions shape survival. And if you've ever wondered what they look like, you'd need a microscope, which is exactly why scientists spend so much time peering through one. Microbes are everywhere in our lives, and they play very different roles. Some cause disease, and those are the ones we usually hear about. But countless others are incredibly useful. They decompose organic waste, recycle nutrients, and even produce energy through photosynthesis. They're also central to industry and food. Without microbes, we wouldn't have vinegar, cheese, bread, or fermented drinks. In factories, microbes are engineered to produce enzymes like cellulase. And in medicine, they've given us insulin and other treatments that millions of people depend on every day. So while some microbes make us sick, many more keep the world running smoothly, working behind the scenes in nature, in our food, and even in our bodies. Microbes connect to so many parts of daily life. In food, for example, bacteria like lactococci ferment milk and give us products such as yogurt and cheese. In water treatment, bacillus species break down organic waste efficiently without producing strong odors, and they do this safely since they're non-pathogenic. Some microbes even generate energy. Certain species produce methane gas, which can be captured and used as fuel. In laboratories, microbes are studied and applied in research, biotechnology, and medicine. But the same microbial power can also be harmful if misused. Anthrax, for instance, is caused by the bacterium Bacillus anthracis. It forms tough spores that survive in soil for decades and can cause serious disease if inhaled or ingested, which is why it has been used as a biological weapon. So this slide is showing us both sides. Microbes as helpful partners in food, energy, and science, and microbes as potential threats if they fall into the wrong context. In agriculture, microbes play an essential role in the soil and inside animals. Some bacteria can capture nitrogen from the air and convert it into ammonia, a form plants can use. This is nitrogen fixation, and without it, soil fertility would collapse. Other bacteria like Nitrosomonas and Nitrobacter complete the nitrification process by changing ammonia into nitrates, which are easily absorbed by roots. Inside animals like cows and sheep, rumen microbes help digest tough foods such as grass and hay. Microbes also recycle key nutrients like carbon, nitrogen, and sulfur, keeping ecosystems balanced. On the other hand, not every microbe is helpful here. Some cause diseases in plants and livestock, leading to losses for farmers. Agriculture, then, depends on encouraging the beneficial microbes while controlling the harmful ones. Microorganisms give us food security and variety. They prevent spoilage in certain preserved foods, like tempeh or salted fish, and they're essential in producing bread, cheese, wine, and many other products. Beyond the kitchen, microbes contribute to energy. Some produce methane, which we capture as biogas, and others, like yeasts, ferment sugars into ethanol, which can be used as a biofuel. Microbes also help clean our environment through bioremediation, breaking down pollutants such as oil spills or toxic waste. Looking forward, the future of microbiology lies in genetic engineering. Scientists are already reprogramming microbes to make medicines, create biodegradable materials, and even clean up heavy metals from water. This slide is a reminder that microbes aren't just part of tradition. They're also key players in future technology. Of course, microbes are not always beneficial. Many cause disease in humans, animals, and plants. Some invade tissues directly, some release toxins, and others disrupt normal body functions. They spread through food, water, air, and sometimes insects. Examples include bacterial infections, viral illnesses, fungal diseases, and parasitic infections. But we are not powerless against them. Hygiene, sanitation, clean drinking water, and vaccines help prevent the spread. Antibiotics and other medicines allow us to treat many of these infections, though resistance remains a growing challenge. This slide reminds us that while microbes give us food, energy, and medicines, they also demand respect because of their ability to cause harm. Here's a set of questions worth reflecting on. How can microbes benefit us in food, in the environment, in preventing disease, in agriculture, in energy, and in wastewater treatment? They're involved in every one of these areas, often in ways we don't see. They feed us, clean for us, protect us, and sometimes make us sick. The challenge for the future is learning how to maximize their benefits while minimizing their dangers. Microbes may be invisible, but their influence on our world is enormous. Let's start with how scientists name and organize tiny life. Carl Linnaeus gave us a simple system called binomial nomenclature. That means every organism gets two names. The first name is the genus, and the second is the specific epithet, which together make the species name. We use this system so everyone around the world talks about the exact same organism. Later, you'll hear rules about capitalization and formatting. But for now, remember, genus first, specific epithet second. Together, they're the full scientific name. Scientific names follow a few tidy rules. They're usually in Latin or a Latinized form, and they're either italicized or underlined. The genus name is capitalized, but the specific epithet is always lowercase. For example, Escherichia coli, which we shorten to E. coli in conversation. That genus, Escherichia, honors the scientist Theodor Escherich, and the word coli points to the colon, the habitat where it's commonly found. Another example is Staphylococcus aureus. Staphylo means clustered, and cocci means spherical, while aureus means golden, referring to the gold-colored colonies it often forms. Scientists sometimes name species after people or places, which is why you'll see names that sound like people's names. Classification is the way we group living things from very broad categories down to specific species. At the very top, we now use three domains: bacteria, archaea, and eukarya. Below domains are levels like kingdom, phylum, class, order, family, genus, and species. Think of it as a nested filing system. Microorganisms are not a single taxonomic group. They're a practical collection that includes bacteria and archaea, plus microscopic eukaryotes such as fungi, protozoa, and algae, and even viruses. So when you hear microorganisms, know it's a mixed group made of several very different branches of life. Let's look at bacteria and how they differ from their look-alike cousins, the archaea. The singular word is bacterium, and the plural is bacteria. Bacteria are prokaryotes, single cells without a true nucleus, and most have cell walls made of peptidoglycan. They commonly reproduce by binary fission, a simple split in two, and they get energy in several ways, by consuming organic or inorganic chemicals, or sometimes by doing photosynthesis. Archaea are also prokaryotes, but they lack peptidoglycan in their cell walls and often live in extreme environments. Examples of archaeal groups are methanogens, which produce methane, extreme halophiles that tolerate high salt, and extreme thermophiles that thrive at very high temperatures. So structurally similar, but biochemically and ecologically different. Eukaryotic microbes cover a lot of ground, so here are two main types to keep separate. Protozoa are single-celled eukaryotes that typically feed on organic matter. They either absorb nutrients or ingest food particles. Many protozoa move. Some crawl with pseudopods like an amoeba, others swim with cilia or flagella. Algae, on the other hand, can be single-celled or multicellular. They are eukaryotes with cellulose in their cell walls, and they make energy by photosynthesis. That means they produce oxygen and organic compounds, and they're a key part of aquatic food webs. So protozoa are mostly tiny consumers. Algae are mostly tiny producers. Fungi are a whole group of eukaryotic organisms, which means their cells have a true nucleus. Their cell walls are made of chitin, the same tough material you find in insect shells. Fungi get energy by breaking down organic material, so they don't photosynthesize like plants. There are two forms to remember. Molds and mushrooms are multicellular and form a network called a mycelium, which is built from thread-like filaments called hyphae. Yeasts, by contrast, are single-celled, tiny one-cell fungi that often behave very differently from the fuzzy molds. Viruses are extremely small, too small to see with an ordinary light microscope, so we usually need electron microscopes to study them. At their core, they carry either DNA or RNA, and that core is wrapped in a protein coat called a capsid. Some viruses also have a surrounding lipid envelope, basically a fat layer taken from a host cell. Important point, viruses can't reproduce on their own. They only replicate inside living host cells. A few special types to know. Bacteriophages are viruses that infect bacteria. Viroids are naked pieces of infectious nucleic acid without a protein coat. And prions are infectious proteinaceous particles, proteins that cause disease on their own. On the other side, multicellular animal parasites, often called helminths, include things like flatworms and roundworms. Unlike viruses, these are large multicellular organisms that you can sometimes see without a microscope. When we talk about cells and microbes, we usually measure in micrometers, also called microns, and written as mim or 1 micrometer equals 1 × 10⁻³ meters. To relate that to things you know, 1 micrometer is 1,000th of a millimeter, so 1 micrometer is 0.001 millimeters, and it's the same as 1,000 nanometers. A few quick size examples to give you a sense. A human hair is roughly 100 micrometers across. A red blood cell is about 8 micrometers in diameter, and many animal cells fall in the 10 to 100 micrometers range. Light microscopes use visible light to make tiny objects visible, and there are several varieties. Compound light microscopes, dark field, phase contrast, differential interference contrast, DIC, fluorescence, and confocal microscopes. Each one emphasizes different features like edges, contrast, or glowing labels. The picture here labels the main parts. The eyepiece or ocular lens is where you look in. The body tube holds the lenses, and the revolving nosepiece carries the objective lenses, low to high power. Objectives are the close-up lenses. Common powers are 4 times, 10 times, 40 cabs, and 100 x oil. The stage holds the slide, with stage clips to keep it steady. The diaphragm controls how much light reaches the specimen, and the light source sits underneath. Use the coarse adjustment knob to bring the image into rough focus, then the fine adjustment to sharpen it. The arm is what you hold when you carry the microscope, and the base supports everything. A compound microscope makes an image in 2 steps. The objective lens first creates a magnified image of the specimen, and then the ocular lens, the eyepiece, magnifies that image again. Total magnification equals objective magnification multiplied by ocular magnification. For example, a 40 times objective with a 10 times ocular gives 400 times total. Resolution is the ability to tell 2 very close points apart. Unlike magnification, resolution determines how much real detail you can see. Modern light microscopes typically resolve details down to about 0.2 micrometers, that's 200 nanometers, under ideal conditions. Shorter wavelengths of light give better resolution, and the refractive index, how much a medium bends light, matters a lot. At high magnification, light can bend as it passes from glass into air and miss the objective. That's why oil immersion is used with high-power objectives. Immersion oil has a refractive index similar to glass, so it keeps light from bending and improves resolution. Let's walk through the main kinds of microscopes you'll meet in a lab. A compound light microscope, the one you usually see in classrooms, uses a stack of lenses plus visible light to make tiny things look bigger. The lenses bend or refract light so the specimen appears closer. A stereoscope, often called a dissecting microscope, gives a binocular, three-dimensional view and is great for bigger specimens like insects, plant parts, or anything you want to handle while you look. The spinning, looking microscope pictured at the top of the slide is an example of this type. Scanning electron microscopes, SEMs, don't use light at all. They scan a specimen's surface with electrons and can reveal surface detail at extremely high magnification. The slide mentions magnifications up to around 2 million times. Transmission electron microscopes, TMs, also use electrons, but they send electrons through very thin sections so we can see internal structures. To make contrast, those sections are often stained with heavy metal salts that block or scatter electrons. In short, light microscopes for routine work, stereoscopes for 3D, SEMs for surface ultrastructure, and TMs for internal ultrastructure. When you pick up a microscope, a few key parts do most of the work. The eyepiece, or ocular lens, is what you look through. Common eyepieces are 10 dimes, though 2 chems or 5 chems exist, and many eyepieces are removable, so you can change magnification. The objective lenses are the main lenses near the specimen, and usually come in a range such as 4 chems, 10 caps, 20 caps, 40 caps, 50 caps, and 100 caps. You rotate the nosepiece to switch between them. The stage is the flat platform that holds the slide. It has a hole so light can pass up through the specimen, and 2 stage clips, one on each side, hold the slide steady. Under the stage, you'll find a diaphragm or condenser that controls how much light reaches the specimen through that hole. Changing it changes contrast and clarity. Older compound microscopes use a mirror to direct light into the optics, but many modern microscopes have their own built-in light source. Finally, the coarse and fine focus knobs move parts of the microscope for focusing. Use the coarse knob for big adjustments at low power and the fine knob for delicate focusing at higher powers. Magnification is simply how many times larger something appears through the microscope compared with your eye. Total magnification equals the eyepiece magnification multiplied by the objective magnification. So, total equals ocular times objective. Most eyepieces are 10 times. With a 4 times scanning objective, you get 10 times 4 equals 40 times total magnification. With a 10 times low power objective, you get 10 times 10 equals 100 tens. And with a 40 times high power objective, you get 10 times 40 equals 400 teps. If you use a 100 times oil immersion objective with a 10 times eyepiece, the total becomes 10 times 100 equals 1000 teves. A quick practical tip. Always start with the lowest power objective to find and center your specimen. Then switch to higher powers and use fine focus to sharpen the image. The ability to see tiny life grew slowly over many centuries and changed how people understood biology. Ancient thinkers like Aristotle, 384 to 322 BC, believed life could arise from non-living things, a view called spontaneous generation. By about 1590, Dutch lens grinders such as Hans and Zacharias Janssen had mounted 2 lenses in a tube to make one of the first compound microscopes. In 1665, Robert Hooke published Micrographia, with detailed drawings made through the best compound microscopes of his day. His work helped people see that living matter had structure. He coined the word cell when looking at cork. Antony van Leeuwenhoek, around 1676, became the first person to describe microorganisms he called animalcules. Later advances in lens design by Carl Zeiss and Ernst Abbe in the 1800s, for example, immersion lenses and apochromatic lenses, greatly reduced optical problems like color fringing and improved resolution. And in 1931, Ernst Ruska built the first electron microscope, opening a whole new scale of detail we can now examine. Antony van Leeuwenhoek used a very small single lens, a simple microscope, to examine scrapings from his own teeth and other samples, and he recorded many different shapes and movements. He described rod-shaped forms, labeled A and B on the slide, various movement patterns or motion pathways, C and D, round or spherical forms, E, slightly longer rounded forms, F, and clustered groups of tiny bodies, H. He called these creatures animalcules, and reported them in a letter to the Royal Society dated September 17, 1683. The little instrument he used had a tiny lens mounted in a backplate, a small specimen mount, and an adjustable focusing screw. Simple, but powerful enough to reveal the first descriptions of what we now call bacteria. Have you ever heard the old idea that life can just pop into existence from non-living matter? For a long time, people believed that. It was called spontaneous generation. In 1688, Francesco Redi challenged that belief by showing a simple fact. Rotting meat kept away from flies did not grow maggots. The maggots only appeared when flies could lay eggs, so life was coming from other living things, not from the meat itself. A bit later in 1836, Theodor Schwann helped shape cell theory, the idea that living things are made of cells, and that the cell is the basic unit of life. Then, in 1861, Louis Pasteur used specially shaped glass flasks to show that tiny living things come from other tiny living things in the air, not from the broth itself. Put together, these experiments shifted thinking from magic or spontaneous generation toward the idea that microscopic life has a source and a history. Think of the years roughly between 1857 and 1914 as a boom time for microbiology. People call it the Golden Age because so many basic discoveries happened fast. Louis Pasteur kicked off the era, and scientists soon connected microbes with disease, immunity, and ways to fight germs. Robert Koch proved that a specific bacterium could cause anthrax, and his lab introduced practical tools like agar plates and the inoculating loop so we could grow and study single kinds of microbes. Joseph Lister brought antiseptic surgery into hospitals around 1865 by using phenol or carbolic acid to clean instruments and dressings. In 1884-85, Martinus Beijerinck described infectious agents in tobacco plants, what we now recognize as viruses, and showed that not all infectious things behave like bacteria. Paul Ehrlich introduced the idea of chemotherapy around 1910 with the drug salvarsan to treat syphilis, and a little later, Alexander Fleming discovered penicillin in 1928. All these advances led to practical changes, better sterilization methods, and the everyday use of aseptic technique in labs and hospitals. Picture a glass flask with a long, curved neck. That is the swan-necked flask Louis Pasteur used. He boiled nutrient broth inside the flask so the liquid was sterilized and free of life. As the flask cooled, air could move in and out, but dust and microbes from the air got caught in the low point of the bend. Because those particles settled in the curve, the broth stayed clear and sterile for hours, then days, then indefinitely. If the flask was tilted or the neck broken so the trapped dust ran into the broth, the liquid quickly clouded with microbial growth. Pasteur's clever design made the point loud and clear. Ordinary air does not magically create life. Living microbes floating in dust and droplets are the source. That experiment, done in 1861 with swan-necked flasks, was a decisive blow against spontaneous generation. Stories about contagious disease go back centuries, but scientists began to pin down how microbes actually cause illness. In 1546, Girolamo Fracastoro wrote On Contagion, an early discussion of infectious spread. In 1835, Agostino Bassi proved that a fungus caused a disease in silkworms, the first clear example of a microorganism attacking animals. In 1847, Ignaz Semmelweis dramatically cut deaths from childbed fever by having doctors wash their hands before delivering babies, showing how transmission by people could be stopped. Louis Pasteur proposed the germ theory of disease in the mid-1800s, the idea that tiny organisms cause many diseases, and his pasteurization process was an early practical application. Joseph Lister translated these ideas into safer surgery in 1867 by introducing antiseptics. He sprayed carbolic acid on instruments and wounds and saw surgical deaths fall. Finally, in 1876, Robert Koch grew the anthrax bacterium outside a host and linked it directly to disease, giving strong, repeatable proof that microbes can cause specific illnesses. Robert Koch developed 4 practical criteria, now called Koch's postulates, to test whether a particular microbe causes a particular disease. First, the suspected organism should be found in every case of the disease and not in healthy individuals. Second, you should be able to isolate that organism and grow it in pure culture outside the body. Third, when a healthy, susceptible host is inoculated with that pure culture, the host should develop the same disease. Fourth, you must be able to recover the same organism from the experimentally infected host. In plain words, find the microbe, grow it alone, make an animal sick with it, and then find the same microbe again. These rules guided decades of lab work and led to techniques like staining, agar media, and pure culture methods. Keep in mind, though, that modern microbiology has found exceptions. Some pathogens can't be grown in simple culture, and some people carry organisms without getting sick. So Koch's postulates are an essential foundation, but scientists use them together with newer methods today. When we prepare a smear for staining, as your micro7.ppt notes, think of 2 simple ideas: staining and smearing. Staining means coloring microbes with a dye so particular structures stand out. For example, the cell wall, a capsule, or internal bodies. A smear is just a very thin film of a microbial suspension spread across a glass slide. After spreading, we usually fix the smear by gentle heat or chemical fixative like methanol, so the cells stick to the slide and are killed. That makes them safe to handle and easier to stain. Make the film thin and even. Too thick, and you'll see clumps instead of single cells. Think of dyes like tiny colored magnets. They carry electrical charges that determine where they go. Stains usually involve positively charged and negatively charged parts. A basic dye has a positively charged chromophore, a cation, and so it binds well to negatively charged cell parts like the cytoplasm or cell wall. An acidic dye has a negatively charged chromophore, an anion, and tends to be repelled by the cell, staining the background instead. When you deliberately stain the background rather than the cell itself, that's called negative staining. It's great for showing delicate things such as capsules because you don't need harsh fixation. Common negative stains are India ink or nigrosin. A simple stain uses just one dye, for example, methylene blue or crystal violet, and it's mainly to reveal cell shape and how cells are arranged. Sometimes a mordant is used. That's a chemical. Iodine is a classic example that helps the dye bind more strongly or forms a complex that makes structures easier to see. Gram staining is a differential technique that separates bacteria into gram-positive and gram-negative groups. The basic steps are crystal violet, add iodine as a mordant, decolorize with alcohol, then counterstain with saffronin. Gram-positive bacteria keep the crystal violet iodine complex and appear purple because their thick peptidoglycan traps it. Gram-negative bacteria lose the violet when decolorized and take up the red counterstain, appearing pink. Clinically, gram-positive organisms are often more susceptible to penicillin and detergents, while gram-negative organisms can be more resistant because of their outer membrane. Some stains are designed to pick out specific features. The acid-fast stain targets organisms with waxy, mycolic acid-rich cell walls. Mycobacterium and Nocardia are the classic examples. Those waxy walls don't get decolorized by acid alcohol, so after staining with carbofuchsin, they stay bright red, while non-acid fast cells take the counterstain. Special stains reveal particular parts of cells. Capsule stains usually rely on negative staining, so the capsule shows up as a clear halo. Endospore staining, malachite green with heat, then saffronin counterstain, makes endospores appear green inside red or pink cells. Flagella are very thin and need special mordant-based or silver-type stains to build up and visualize their number and arrangement. And of course, gram staining remains the standard differential test for gram-positive versus gram-negative bacteria. Microorganisms are incredibly diverse, and we study them with a set of characteristic techniques. Good aseptic technique to avoid contamination, pure culture methods to isolate a single species, microscopic observation to see shape and arrangement, and the variety of stains we've talked about to reveal walls, capsules, spores, or special cell features. You might also see terms like psychrophiles. That's just a name for cold-loving microbes. If you want to read more, Prescott's Microbiology 6th edition is a classic reference. Overall, staining and culture methods are powerful tools. They reveal structure, help identify organisms, and explain why some bacteria respond to certain antibiotics while others resist them. And that wraps up this part of our microbiology journey. If you found this helpful and want to keep learning with me, don't forget to hit that subscribe button and turn on the notification bell. That way, you'll never miss the next lesson. And if you enjoyed the video, a like or a comment really helps the channel grow. Thanks for watching, and I'll see you in the next one.